Hello everyone, this is my lab project. We were asked to do 15 worksheets and all explanatory of biosynthetic pathways. We'll be going through most of these worksheets to understand how nucleotides and ribose is produced and how ATP is also produced and directly correlated to DNA replication. So ATP production and regeneration via ATP synthase is going to be explained, as well as the sources for ATP for reaction mechanisms. So we'll start over here on these two worksheets, number 4 and 5. We can see that PRP is a metabolite. It's a metabolite that is directly associated with inosinate. Inosinate, seen here and also here, is directly correlated to ATP production. We can also gain inosinate from meat, like chicken. It's commercially produced usually, types of meat, and it has an umami flavor. With that, we can also come over here to both lactose and sucrose, so sugar, ingestion of sugars. Mostly anything we eat is broken down into a sugar. So sucrose being broken down into glucose and fructose, lactose being broken down into galactose and glucose. That glucose molecule, when metabolized by an enzyme or electron carriers like NAD plus or an enzyme like sucrase, is able to begin a process called glycolysis. Glycolysis is the breakdown of glucose into smaller subunits. ATP, as you can see, is used in this pathway. The mechanism of ATP being used is less so energetically demanding because you're starting with a larger molecule and breaking it down into pyruvate, which is shown here. Pyruvate is a smaller molecule. We are going to produce around 30 to 38 ATP via this mechanism pathway. We are able to synthesize nucleotides. Go this orientation. We're able to synthesize nucleotides by biosynthetic precursors like inosinate, ATP, carbohydrates, the metabolism of amino acids, and ammonia. So all 4 nucleotides are synthesized in liver cells, cytoplasm. This makes them bioavailable. So like you can see, like pyrimidines and purines, like alanine, aspartate, glutamate, these are all broken down and sent into a mechanism called the citric acid cycle, or in the pyruvate dehydrogenase complex. So within these cycles, ATP is, electron carriers are used to generate energy enough for this process to occur. With the generation of ATP and also throughout glycolysis, so not just in those pathways, oxidative phosphorylation is occurring, mainly in the glycolysis pathway. So with the regeneration of ATP, this is the mechanism to do so. We can see that it kind of functions like a rotating wheel, kind of like a child's toy in a way. So the gradient movement across the matrix is what causes these little wheels to turn. There's multi-step binding, and it drives that rotation. So phosphate is released and docked, and released and docked via hydrolysis, and it causes a proton pump to pump hydrogens across a matrix, and this is the mitochondrial matrix that we can see here. This is where the electron transport chain occurs. To continue, the nucleotides, so the nucleotides like pyrimidines and purines. Purines are formed via a sugar template. We'll go back to that worksheet here. The pyrimidines, like aspartate and carbamoyl phosphate, are really integral to DNA replication. We also metabolically parallel to glycolysis can see that during the pentose phosphate pathway, as depicted here, is associated with ribose, and ribose is made from glucose. So this generates NADPH, which is a precursor, and pentoses, or 5-carbon sugars. So R5P is really important, as depicted here. So ribose 5-phosphate is a very, very important precursor for nucleotide synthesis. So we can see that during the metabolism of PRPP, and also like shown here, and also the use of it from the 5-carbon chain into fructose 6-phosphate. So yeah, fructose as well as glucose is really important to our cells' livelihood. We need these nucleotides to be sourced from glycolysis mainly, and the pentose phosphate pathway to meet our needs of aerobic respiration. So all of these little integral metabolites, precursors, energy sources really are required for DNA synthesis and for life in general. So I hope you enjoyed my presentation. I wish you all luck on the final, and yeah, thanks.